this dip right here, and then you see how a little bit of cerebellum's bunched up here. These are called the tonsils, and they are just a landmark. They are really components of both the spinal and the cerebrocerebellum, but their clinical significance is that if they herniate through the foramen magnum, and that's what's causing this groove, is that it kind of sets up on the bony prominence of the inside of the foramen magnum. If they herniate through there, then they start to pinch the brainstem. And pinching the brainstem then causes all kinds of clinical problems because you're getting cardio and respiratory centers shutting down, okay? But if you look out laterally, kind of at the interface between the pons and the medulla, you find this other small group of cerebellum that's kind of divided off separately. This is the flocculus. And that flocculus, as an anatomical structure, is a part of the vestibulocerebellum. So this part of the cerebellum is what starts to drive or influence vestibular nuclei and vestibular function. Okay? Everything else, all of this out here, as the hemisphere of the cerebellum, is the cerebrocerebellum. So that's the part that has to do with sending information to cerebral cortex, affecting lateral premotor cortex to help facilitate and sequence and fine motor activity, putting fractions of movement together into what we see as movements. Okay? Okay, so let's put this guy right here. Now we're going to do the same thing, is we're going to take and start at the interventricular foramen, and what I'm going to use as a way of identifying this is I'm going to use the uncus, or that part of the temporal lobe that overlies the amygdala. I'm going to use that as a way to define coronal. Okay? So either there, maybe I'll do it this way so everybody can see. There. Here's the bump of the uncus right here. That's oculomotor nerve right there coming out of interpeduncular fossa. And you see how there's kind of a bump right there? That's the uncus. Okay? So then I'm going to take that and that, and I'm going to make a nice cut. Now, think about this. I made a cut at interventricular foramen up and down like this. So I should then be able to look from medial to lateral and find the genu of the internal capsule, the globus pallidus, the putamen, the insula, lateral sulcus, and then temporal lobe. Midline, internal capsule coming all the way to the midline. Globus pallidus, this one you can actually see both sections. Putamen, insula, lateral sulcus. Okay, so I've made that cut right through there. Now, this is the insula.
Would that be the posterior limb of the internal capsule then?
At this point, it's difficult to define because remember that the internal capsule forms this fan shape going up, so we don't really define it. It's probably mostly stuff coming from parietal lobe and occipital lobe, so it's probably more posterior limb than anterior limb, okay? But remember we cut through the uncus. And here we are at the anterior part of temporal lobe, and now you can see in coronal section the look of the amygdala. Here it is horizontally, here it is coronally. Okay? This little structure right here is the optic tract as it sweeps around the brainstem. Okay? What else do I want to talk about there? Okay, so now what I'm going to do is I'm going to make a cut back here. So like back caudally to that. So I'm going to cut through the thalamus I'm going to cut through the posterior limb of the internal capsule. I might get the caudalmost part of the putamen, get the caudalmost part of the insula, and go back. But what I will get with this cut is I will be cutting through the brainstem in somewhat of a tangential section that goes through midbrain, through pons, and just maybe the very beginning of the medulla. Okay? So I'm going to come back here. No, I'm not going to get medulla. I'm going to make a cut in front of that. So I'm going to make this cut. Go to thalamus. Here's the internal capsule. Now I am in the posterior limb. Here you can see a little wedge that is only just putamen. There's no more globus pallidus in it because I cut way back here. Okay? What you can now see is what I asked you to try to imagine over here, as you can now see the continuity of the posterior limb of the internal capsule as it now becomes the cerebral peduncle from here to here. And then those fibers begin to break up into those corticospinal fibers that pass through the ventral part of the pons. And then they're going to all collect again right here as the pyramid. So you can see the continuity of that entire corticospinal pathway till it gets to the cord. Okay? You can also see on this, again, you can see pars compacta, the dopaminergic cells of substantia nigra. And you can see kind of this circular area right here. That's red nucleus. Okay?
Is pars compacta this right here too?
Yep, just the black right there. Now, remember what I did over here is I pointed out the optic tract. Remember, if you look at slide 9, it shows the optic tract skirting around the lateral side of the brainstem, of the midbrain, of cerebral peduncles, and ending in lateral genicular nucleus. So we're going around the cerebral peduncles, and we get all the way back to the edge of the cerebral peduncle, and right there is lateral genicular nucleus. You kind of see this upside down little hat, or Napoleon's hat right there. That's the lateral genicular nucleus. Okay?
If you said that's sometimes gold color?
Yeah, sometimes it shows up more gold than on this one. Okay?
Is that the optic tract right in front of it?
No, this is actually, there's a little loop in the hippocampus as it forms, and that's what that is right there. But what you can see is this is more consistent with the images that I've shown you of the hippocampus. Here's perihippocampal gyrus, and hippocampus is folded up inside of that. Okay? So folded up inside of that, it's a series of two C's kind of interlocking. And the output of this, and this is where this is a little confusing, this little flop here, but the output of this fiber bundle right here that's hanging on to that little recurved part of the hippocampus is the fornix. Okay? Remember, the fornix is right here. The fornix doesn't stay all on the midline. It starts out laterally in temporal lobe and sweeps around posteriorly and then shows up superiorly right here. And if we followed this around, can't do it very well. If we followed this around the brainstem, right there is the fornix. So the tip of the probe is coming out around the brainstem, the superior side of it, to become the fornix. Okay? Now, if we do the same thing and we go anteriorly, so anteriorly, if we go anterior to this, we're going to make a cut across the ventricle. Remember, here's the interventricular foramen right here, where the genu is, at the front part of the thalamus. We're going to cut through the ventricle. We're going to cut through the head of the caudate nucleus, through the anterior limb of the internal capsule, through putamen, and again, probably missing most of globus pallidus, and then out to the insula and lateral sulcus. Okay? do this, I usually lose temporal pole. Okay? So remember, cut through lateral ventricle, caudate, anterior limb, and putamen. Here's septum pellucidum, here's lateral ventricle, here's the caudate nucleus, here's the anterior limb of the internal capsule, here's the putamen. And now we're out here, we put temporal pole back up there, like that. It should be setting like that. Because now we're at the front part of the insula, okay? And the very front part of the lateral sulcus, all right? So, your task is when you take these and you put them back together, is to look at them and kind of identify sort of how things are moving. I will let you know that the first question somebody asks with respect to the test is, are you going to have these laid out like this so I can see orientation? And the answer will be no. Sorry about that. But the next thing is how will I know whether I'm looking at the top of this or the bottom of this? And it doesn't really matter. Because in reality, if we put these together like this, they become mirror images and you really see that bilateral look that I showed you in class. So it doesn't really matter. Okay? So that really becomes what you saw in class as that image horizontal that we then use to describe internal capsule, lentiform nucleus, and that kind of stuff. Okay? So do you have any questions?
Superior peduncles again. Can you identify the superior, middle, and inferior?
Not on this cut. I can do it for you, but then I'll have to kill you.
I'm okay with that.
So.
Yeah, kill me.
Do it now before the test, huh? Okay. Now, if you think about slide number 4, and you have kind of Mickey Mouse ears out here on the side of the inferior cerebellar peduncle, which is pulling all this afferent information in. And remember that it's kind of setting just off to the side of the dorsal columns, the other side, just to the other side or the outside of dorsal horn. Well, look what we have here. Here are all these dorsal roots coming into the spinal cord. Okay? So here are all the dorsal roots coming into the spinal cord. That means if we went just underneath them, dorsal spinal cerebellar tract would be running right along there. Okay? If we kind of pull this away, you see how that area then kind of bulges up right here as it starts to collect more and more fibers, vestibular fibers, olivocerebellar fibers, and that's the inferior cerebellar peduncle from the outside. Okay? See this ridge right here. Okay? The middle cerebellar peduncle are all these fibers of the transverse fibers of the pons that are all coming from the contralateral side, and they're all collecting into this point. And right here you see coming out of the side of that is the trigeminal nerve. Okay? Now the superior cerebellar peduncle is much harder to show, but you can kind of get a sense if you look at the inside of the fourth ventricle, and you have this component right here coming down out of the superior cerebellar peduncle. And when I say coming down and coming out, is I'm going back to the generalization that the inferior and middle cerebellar peduncles are primary afferent pathways carrying information into the cerebellum, and that the superior cerebellar peduncle is the primary pathway carrying information out. Remember that that is not completely true. But the superior cerebellar peduncle forms this dorsal part of the roof of this fourth ventricle, and as it comes down and you can kind of see a little bit of white right here.